Hello everyone and welcome back to Cell and Gene Therapy series on Only Pharmacy. In our previous lecture, we introduced the field of cell and gene therapy and understood how these technologies are changing modern medicine. In today's lecture, we are going to focus on one of the most important areas of regenerative medicine, stem cell therapy. So what exactly makes stem cells special? Unlike most mature cells in a body, stem cells have two remarkable properties: self-renewal and differentiation. This means that they can maintain their own population while also producing specialized cells such as blood cells, bone cells, muscle cells, and potentially many other cell types depending on their potency. Because of these properties, stem cells have attracted enormous interest in regenerative medicine, tissue engineering, drug discovery, and disease modeling. But an important point is that not every stem cell can form every type of tissue, and not every stem cell treatment advertised commercially is scientifically or clinically established. So in this video, we will understand the science behind stem cells, their different types, how stem cell therapy works and where it is actually being used. Let's start with the fundamental question: what exactly is a stem cell? Stem cells are essentially undifferentiated cells that have the ability to produce more stem cells and depending on their potency differentiate into specialized cell types. The first important property is self-renewal. Self-renewal allows a stem cell to divide while maintaining the stem cell population. This is essential because tissue that continuously require new cells one need a reliable source of replacement cells. The second property is differentiation. During differentiation, a stem cell becomes more specialized cell. For example, hematopoietic stem cell in the bone marrow can give rise to the different blood cell lineages. This leads to an important concept: potency. Potency describes the developmental potential of a stem cell, essentially how many different types of cell it can generate. At the highest level, we have totipotent cell, followed by pluripotent, multipotent, oligopotent, and unipotent cells. So stem cells shouldn't simply be thought of as a universal replacement cell. Their therapeutic potential depends on their source, potency, biological properties, and the disease being treated. This distinction is particularly important when we hear claims about stem cell being able to regenerate any organ or cure multiple unrelated diseases. Now let's look at how stem cells are actually classified. Stem cell can be classified into several ways, but two useful approaches are classification based on potency and classification based on source. Based on potency, we have totipotent, pluripotent, multipotent, oligopotent, and unipotent cells. Totipotent cells have the greatest developmental potential. Pluripotent cells, such as embryonic stem cells, can differentiate into cells derived from all three embryonic germ layers. Multipotent cells have more restricted potential. A good example is hematopoietic stem cell, which generate multiple blood cell types. Now let's consider the source. Embryonic stem cell or ESC are derived from the inner cell mass of the blastocyst and are pluripotent. They have enormous research potential, but their use raises ethical consideration and concerns such as tumor formation. Then we have adult or somatic stem cells. They are found in tissues such as bone marrow, adipose tissue, and other organs. One of the most clinically established examples is the hematopoietic stem cells. We have we also have the perinatal sources including umbilical cord blood and cord tissues. And finally, there are induced pluripotent stem cell or iPSCs. These are particularly fascinating because a mature adult cell can pre-program back into pluripotent state. This technology opens major opportunity in disease modeling, drug discovery, and potentially personalized regenerative medicine. And now let's move from classification to the actual therapy. So, how does stem cell therapy actually work? Although the exact mechanism depends heavily on the cell types and disease, we can understand the general process in several stages. First is cell sourcing and isolation. Cells may be obtained from sources such as bone marrow, peripheral blood, and other appropriate tissues. The cells are then processed. Depending on the therapy, they may be purified, expanded, differentiated, or genetically modified before administration. The next stage is administration. This could involve the infusion into bloodstream or delivery to a specific anatomical site, depending on the therapeutic strategy. Once administered, some cells may undergo homing or migration toward the particular tissue. But here is an important scientific point. Stem cells do not necessarily work simply by replacing the damaged tissue and becoming new cells. Their therapeutic effect can also involve the paracrine signaling and immunomodulation. In other words, cell can release signaling molecule and growth factor that influence the surrounding environment, inflammation and immune response, and tissue repair. For some therapies, engraftment and long-term repopulation are critical. For other, the therapeutic effect may be strongly associated with signaling and immunomodulatory mechanism. Therefore, mechanism is therapy and disease dependent. This is one reason why understanding the specific cell product is so important when evaluating a stem cell therapy. Now let's see where these therapies are actually being used and where the field is heading. The most established clinical application of stem cell therapy is hematopoietic stem cell transplantation, commonly associated with bone marrow transplantation. It has become an important treatment option for conditions such as leukemia, lymphoma, multiple myeloma, aplastic anemia, and selected inherited blood disorders. Beyond hematology, researchers are investigating stem cell approaches in orthopedics, neurology, cardiology, ophthalmology, autoimmune diseases, and many other areas. However, it's important to distinguish between established clinical treatment and experimental or investigational applications. This distinction is extremely important because of the stem cell therapy is sometimes used commercially to promote treatment that do not have strong clinical evidence. These are also significant challenges. These include safety, tumorigenicity, immune compatibility, limited cell survival, manufacturing complexity, cost, storage, and regulatory requirements. For a cell therapy to become successful medicine, it isn't enough for the cells to show promising biological activity. We also need a reproducible manufacturing process, appropriate quality controls, consistent potency, acceptable safety, and convincing clinical evidence. Looking toward the future, several technologies could transform this field. Gene editing may allow scientists to modify stem cell before therapeutic use. iPSC technology could enable patient-specific cell model and potentially new regenerative therapies. Organoids and tissue engineering may help recreate more complex human tissues, and technologies such as 3D bioprinting and AI assisted manufacturing could further expand the possibility of regenerative medicine. So, the key message from the today's lecture is simple. Stem cell therapy is not just about replacing damaged cell, it is about harnessing the biological property of cells to modify disease, restore function, and potentially regenerate tissues. At the same time, successful stem cell medicine requires rigorous science, manufacturing, clinical evidence, and regulatory oversight. That brings us to the end of the lecture 2. If you found this video useful, don't forget to like, share, and subscribe to Only Pharmacy. In the next lecture, we'll discuss one of the most exciting example of engineered cell therapy, CAR T cell therapy, including its mechanism, manufacturing process, clinical application, and challenges. Thank you for watching, and I'll see you in the next video.